Ladies and gentlemen, dear colleagues and Marsilius fellows, and in particular, dear Donald Ingber, I would like to welcome you all to today's Marsilius lecture for the summer term 2023. We are honored that so many of you have decided to come to the Aula, the old auditorium of our university this afternoon, to join us for a lecture that covers the boundlessness of creativity. Creativity knows no boundaries. This could be the theme of today's lecture by Donald Ingber, whom I would like to welcome here today. He will tell us in a moment how different disciplines, different impulses from art and design have influenced his thinking about biomedicine and cell mechanics. And he will take us on a journey into thinking about scientific questions in a highly interdisciplinary manner, a theme that fits very well what we also like to accomplish with the Marsilius-Kollegs. We are very pleased that he accepted our invitation to come to Heidelberg and agreed to give the 31st edition of our long-standing lecture series. Thank you very much for taking the time and coming here today. I would also like to take this opportunity to thank Frank Wassermann on the grand piano and Vitali Titar on the bassoon, to whom we owe the festive musical framework today. They have just performed for us the Largo from Antonio Vivaldi's Sonata in G minor. The closing piece will be Aria by Jean-Marie Leclerc. Thank you both for this inspiring contribution. My name is Michael Boutros. Together with my colleague, Friederike Nüssel, we are the directors of the Marsilius-Kollegs, the Interdisciplinary Institute for Advanced Study at the University of Heidelberg. Since the founding of the Kollegs, the semi-annual Marsilius lectures have been an important part of the academic year at our university. Outstanding scholars are invited to Heidelberg to give a lecture on a topic that is of interest to a wide audience, also within the academic discourse. These lectures often focus on current grand challenges: climate change, environmental degradation, digitalization and democracy, question on health and participatory justice, new scientific frontiers, and technological revolution or phenomena of cultural change. The lectures stimulate an interdisciplinary exchange at the Marsilius-Kollegs with new ideas and new impulses. They also make an important contribution to the public communication of science at our university. I'm very confident that our speaker today will not be deterred by grand challenges. Donald Ingber is not only a pioneer in biological-inspired engineering. Over the last decades, his work has led to major advances in mechanobiology, cell structure, tumor angiogenesis, tissue engineering, systems biology, nanobiotechnology, and translational medicine, all important areas that impact health and well-being of humans worldwide. We will hear more about his pioneering research and the development of special technologies in his lecture. Donald Ingber's scientific career began at Yale University, where he received a BA, an MA in molecular biophysics and biochemistry, and later on a combined MD, Ph.D. In 1984. He then moved to Harvard as a postdoctoral fellow, working in the department of the founder of angiogenesis research, Judah Folkman. He remained at Harvard, was appointed to a professorship of pathology in 1999, and in 2002 was appointed senior associate in the vascular biology program at Boston Children's Hospital. Since 2004, he held the position of Judah Folkman Professor for Vascular Biology at Harvard Medical School. In 2008, he became Professor of Bioengineering at the Harvard School of Engineering and Applied Sciences, in addition to his professorship at Harvard Medical School. A year later, he founded the Wyss Institute for Biological-Inspired Engineering at Harvard, which he still directs with great success. And we've just heard during lunch on many kind of inspiring ideas how you structure interdisciplinary environment. Donald Ingber has done groundbreaking work on mechanical forces and its role in many biological processes. But also let me give just you a few figures of the success of the Wyss Institute. As of today, more than 2,800 publications have been published, more than 4,000 patents have been registered, more than 120 commercial licenses, and 55 startups fall into his time as director. I think these are very, very impressive benchmarks. I would spare you to listing all prizes and awards that Donald has received over the year, just to mention his appointments to all the 3 national academies, including Academy of Engineering, Medicine, and Inventors, a Breast Cancer Innovator Award of the U.S. Department of Defense. Last but not least, and I think that's also particularly interesting for today's talk, Donald Ingber's worldwide collaboration with artists, architects, and designers have resulted in the exhibition at the MoMA in New York, the Design Museum in London, the Gropiusbau in Berlin, and the Centre Pompidou in Paris. I think you will all agree we could have found hardly anyone better suited to talk to us today about bridging the gap between science, engineering, and the art. Dear Don, we are very excited to look forward to your lecture, and thanks again for joining us here today.
Thank you for showing up in the heat. It'd be nice if there were a pool at the end, but otherwise, thank you so much for inviting me for this incredible honor and this incredible lecture room. So what I'd like to do in the next 50 minutes or so is sort of take you through my path to how I ended up crossing barriers between science, engineering, and art. And when I started as a student, I was always interested in the fundamental question of how living cells and tissues are constructed. And literally in the 1970s, I started to propose the idea, which is sort of heretical at the time, that mechanical forces may be as important as chemicals and genes. And this, as I think many of you know, is now a major tenet of the burgeoning field of mechanobiology. But how did I get there, and how did this all happen? Well, a good question. It actually all started when I was an undergraduate science major, and I decided to take an art class at Yale University, where I was at the time. And I don't know how it is here, but I even sense it is here already today, that the science students are on one side of campus and the art students, humanities, on the other side of campus. But one day, I was taking courses in molecular biology, which in the 70s was before cloning, so it was the biophysics of molecules, really. And I saw an art student walking around with a sculpture that looked exactly like my virus, and I started to say, what class are you building this in? And he said the class was called three-dimensional design, and I was learning that everything at the molecular level is three-dimensional design. The shape of the helix of DNA is critical for its replication, and actomyosin, how they interact and twist and create forces. And so I took this course, and in this course I was introduced to a form of architecture that I had never heard about called tensegrity. Comes from tensional integrity. Now, most buildings, like this building, are built by sort of laying one building component, let's say one brick atop another, and they're stable, but if it's hit by a truck, it falls to the ground. Tensegrity structures use, instead of continuous gravity-based compression, use continuous tension. And this actually came out of the Buckminster Fuller's geodesic dome work, where he tried to figure out why do these get more efficient as they get bigger, and he realized that you could actually replace struts with strings and you still have these sorts of structures. And artists have described the human body this way. If you go to the anatomy lab, you will see a skeleton, and it looks like us, but it has to be wired together and hung from a stick. In reality, we're pulled up against the force of gravity by a continuous series of tensile muscles, tendons, and ligaments that basically tense our body and create stability. So my arm can be floppy or it can be stiff, and that's all due to internal tension. And that's what's called pre-stress in engineering, and that is a fundamental property of these sorts of systems. Now, in this class, that's my sculpture on the left, which I still have. The teacher had a model with elastic elements one day, I'm sorry, and he was talking, and as he was talking, he was pressing it up and down, and then he let it go, and it bounced up into the air like this. I have a slow motion for those of you who didn't see it in the back. And it was all because of that internal tension. Nice catch, isn't that very good? Anyway, so in probably the first really serendipitous experience of my life, 5 days before, I had been learning how to culture cancer cells in a cancer research lab on the other side of campus. And when you trypsinize cells, which clips their anchors to the dish, they spontaneously round up just like that. And I thought, oh, cells must be built like tensegrity structures, because about a year or two before, the first papers came out that all cells have an internal molecular framework known as the cytoskeleton or cell skeleton. These are nanometer-sized cables and filaments that are much like in muscle, but it wasn't until the mid-70s that people realized that all cells, even immune cells, nerve cells, have these contractile proteins that create tension. And so in the last chapter of my PhD thesis, for those of you who are graduate students, I actually built models out of sticks and strings. The sticks are aluminum struts I stole from the chemistry lab that would orient glassware. The cables are shock cord from a sailboat, and I built the nucleus and attached it to the top of the cell by black cord. You can't see it, but when I spread the cell, the nucleus spread, and when it rounded, both rounded in a coordinated manner. And I'll come back to that in a few minutes. Now, same time I was taking art and molecular biophysics, I was taking developmental biology, and there I would see movies of embryos. This is zebrafish. The yolk sac is at the bottom. These are the cells. And you have 2 cells, and then 4, and then 8, and then 16, and 32, and 164, and so on. And then they hit a point where they just start pulling on each other and moving and physically sculpting all the organs. You can see all the forces are generated by the cells in that actomyosin cytoskeleton. So everything that you learn about in developmental biology and cell biology is happening in an incredibly mechanically active environment. Furthermore, if you take cells out of tissues and you put them on a dish, they spread. But if you put them on a flexible substrate, almost like a paint film drawing, they pull it up into wrinkles. And that's because cells form spot weld-like adhesions called focal adhesions, and actomyosin filaments pull them together. And it's the ability of the dish to resist the pull that lets the cell spread itself outwards. About 2 years later, Judah Folkman published in Nature a paper that's a classic, which said that it gave evidence to suggest that the shape of the cell could dictate whether it can grow or not. You give it all the growth factors you want. When it was spread, it would proliferate. When it was round, it would stop. And I was interested in how tissues are constructed. And they are constructed usually to create fractal-like forms by repetitive bending or branching. And they're usually localized regions of growth that grow out more, and then the sides of them grow, and that's how you get differential fractal-like patterns. But I even started to think of this mechanically. And so if you have a cross-section to an epithelial tissue, a lining tissue, these cells, red, are going to grow, and that's going to give you the bud. But what people found, Mert Bernfield did this in the late 70s, is that before you get growth in this spot, there's a local thinning of the scaffold beneath called extracellular matrix, or basement membrane. Think of it like an egg carton on which cells sit on. But that egg carton is under tension, and so I thought, much like a run in a woman's stocking, that if the matrix is under tension at all times, and I should say, I know from medical school that all of our tissues are under tension. That's why a surgeon has to suture a wound together, because it's pulled apart. Or if you cut your muscle, it's pulled off the bone. So if that's under tension like a stocking, then if there's thinning of it, which is done enzymatically, that will stretch out more, and that will cause the cells to stretch. And if Judah Folkman were right, that would give you local growth. And if you were to do this in a repetitive way, you'd get patterning. But I also realized that in medical school, cancer, people see that there's thinning of the extracellular matrix before you get cancers forming, and in the regions that they will form. And so the idea here was, in the embryo, you get thinning and through degradation, but you put out more new matrix, which is how you build this bud. It has more total matrix. But in cancer, you can't keep up with the degradation, so you get the growth, the stretching and the stimulation, but you don't get the outpocketing. Instead, you get a pile. And to shorten to the cut to the chase, we and many others have shown that this is exactly what happens over the last 30 years. Now, I went to Judah Folkman's lab to do a postdoc when I got my PhD because he was working on cell shape. He was also the founder of tumor angiogenesis, the idea that cancers can't grow past about 1 millimeter in diameter without oxygen, and they need to get new blood vessels to form. And then I had my second serendipitous experience in science. I'm a very lucky person, or else at least I act when I see things, but I had a fungal contaminant in my culture, which normally everyone says, quick, put it in the autoclave, give it, iodine solutions, kill it so we don't spread it. But I saw that in my culture, instead of all the cells popping off the dish, there was a gradient of cell rounding so that right beneath it they popped off the dish, and then they were round, and then they were spread, looking fine a few diameters away. So I thought, maybe this produced a factor that caused cells to round. These were capillary blood vessel cells. Maybe I had something that would inhibit capillary growth. Maybe it could be useful for cancer. Another long story short, that eventually went to clinical trials. It was developed by a company called Takeda in Japan. And it's one of the most ubiquitous inhibitors of angiogenesis. It led to a complete remission in the New England Journal, but it had some neurotoxicity. The company also had some financial challenges, and so that stopped. But there's now a new version of this that's in phase 2 clinical trials that doesn't cross the blood-brain barrier, and I'm hoping we'll see something really coming out of it soon. But this introduced me as a scientist into the world of patents and clinical trials, which you don't get as a basic scientist. Around the same time, some of you may have heard of Bob Langer, who's one of the most productive engineers in the world and startups in the world. And he was in the same department. He was also a postdoc with Judah Folkman, although a little bit ahead of me. And in the late 80s, we formed a tissue engineering company called Neomorphics, where we were developing biodegradable polymers mixed with cells to create artificial tissues, which to this day is still the core technology behind the field. And through that, I learned about biotechnology startups, and which is a whole other lesson. You don't have impact unless you figure out how to commercialize your discovery, and that's something I think is not taught to graduate students in science. But I was still very interested in this idea, does mechanically distorting cells really control its function? And people with Folkman's paper said that it could be how many adhesions the cell make or other things going on. So what I wanted to do is I wanted to make cell distortion an independent variable. I wanted to give them optimal amount of soluble growth factors, which is the way most people thought cells are stimulated to grow, to do it in a chemically defined medium, so I knew everything there, which we did. And then the idea was, if I could make little adhesive islands on the size of individual cells, that if surrounded by Teflon-like non-adhesive regions, then if I put a cell on this matrix, like the egg carton in your body, the same proteins are coated on it, the cell would stick, pull, flatten, and when it came to the non-adhesive Teflon-like region, it would stop because there's no resistance. And I would get, if it's a circle, I get a pancake-shaped cell. If I did the same thing with a smaller island, I get a cupcake, and if I had a tiny island, I get a golf ball on a tee. And the only variable would be distortion. And we did this in one of the earliest demonstrations of sort of nanobiotechnology through a collaboration with George Whitesides, who's world-leading chemist at Harvard. He had developed an inexpensive way to make microchips for the computer industry, and microchip manufacturing gives you control over the same feature size, nanometer to micrometer, that living cells and tissues live at. So in conventional, and this is, by the way, lithography, which artists use to do lithographic artwork. In microchip printing, you take a silicon wafer, you make a pattern on a computer, whatever pattern you want, and you create a pattern using ultraviolet light because you put a mask over the surface that protects some regions, not the others, and you get, let's say, these circular holes in the pattern. In microchips, you do this in a clean room, you etch it, and you get the pattern. What George devised is a technique where you make that, and then you take a liquid material that's called polydimethyl siloxane. It's silicon rubber. You pour it over it, let it permeabilize, peel it off, and you have a rubber stamp, but you retain the resolution of those little nubs down to 60 to 90 nanometers. And then you could take that and you could stamp chemical inks and make thousands of these outside of a clean room. And so this is called microcontact printing or soft lithography. And you coat the intervening surface with molecules that self-assemble into a complete a defined monolayer at the nanometer scale, that you have a molecule, polyethylene glycol, that prevents protein absorption, cell adhesion. And when we did this, and we put, and this is how I got into the world of nanotechnology. When we did this, if we put this capillary blood vessel cell from human on a big island, you got a pancake, small, you get a golf ball on a tee. The reviewers of the paper said, well, there's more total contact or matrix area here than here. So we took this little island and we made many tiny islands, and the cells spread like a suspension bridge from dot to dot to dot. So the cells spread like this, but it had the contact area of that. And with many different cell types, we found that as you increase cell stretching, you get a concomitant increase in growth. And when you rounded them, even though they were still adherent, you turned on a cellular suicide program called apoptosis. And then we made islands. These are capillary blood vessel cells that normally make tubes. And we made lines instead of dots. And now they make tubes with a hollow lumen down the center in vitro, where the same cell in the same medium right next to it with no patterning is growing as a monolayer. And finally, we made islands with square corners, which cells don't normally have. And now we can show that when you give factors to stimulate motility, movement, cells move by putting out little processes called lamellipodia and filopodia that are filled with actin filaments. And you see they only come out of the corners, where in a round cell, there's no bias. And this is because that's where tension is concentrated. Just look like if I took a flexible membrane and stretched it over this podium, it would stretch more in the corners. And that is what guides molecular patterning at the nanoscale, even though forces are at the microscale. And we can confirm this is true in vivo. This is a mouse where we put endothelial cells in a gel of different stiffness, which is different ways to resist cell spreading, and we could show that we can control angiogenesis in vivo. But the fundamental question was really, to me, was how do cells sense and respond to these forces, which people could never really explain. We obviously know when you lift weights, your muscles grow, but nobody really knows how this works. And this was what we called mechanotransduction. How do you transduce a mechanical signal into a chemical response inside the cell? Now, when I got involved in the field, people viewed cells like a water balloon filled with molasses. A lot of people still do. And it was very hard to understand how, pulling on the surface can give you specific changes in the cell. But because of this tensegrity idea, cells really would be thought of more like a tent. If you want to put up a tent, you have a flexible membrane. You put up poles to tense it. You put pegs in the ground. You tie the string to put it under tension, and you have a stable structure. And what this suggested then is that, like a tent, cells would not feel forces the same everywhere on the membrane. It would feel it through the tent pegs in the ground. And so to pursue this, I now had to reach out to collaborators in fields like physics, engineering, because this is not cell molecular biology type of questions. And in 1993, we developed a technique called cell magnetometry, where we took magnetic beads that were anywhere from 30 nanometers to 5 micrometers. We coat them with specific molecules, so they bind specific receptors on cells. And then we could apply magnetic forces to either pull them away or twist them, and we could measure how far they move or twist. So we could do stress-strain curves, which is an engineering approach to look at material properties. This is a cell on its side. It's actually growing on a wire. And this is the nucleus. That's the nucleolus, and this is a bead. And cells are incredibly strong. This is nanometer-level forces. What we found here, though, is that if we pulled on transmembrane receptors that are all over the cell, but they're not involved in adhesion, they're not tent pegs, cells are very floppy. But if you pull on integrin receptors, which are receptors that cells use to bind to that egg carton or matrix, cells get stiffer and stiffer as you pull on them in direct proportion. And this is a paper in Science, and it's 30 years ago to the year. We were able to show, I took the nucleus out of my cell model, hung it from a stick, put weights on it, and I showed we got exactly the same linear stiffening response that we saw in cells. I could also use drugs to show that all 3 cytoskeletal filament systems contribute to this. This just wasn't actin or microtubules or intermediate filaments. We also could show that pulling on integrins, but not other receptors, activates chemical signaling inside the cell all the way to gene transcription. And I think I have one picture of this. That's the bead that we're pulling on. And at the right, you're seeing calcium imaging. The bead is at the cell surface, and these little lightning flares, solar flares, are calcium going into the cell with each pull. And this is in 5 milliseconds. So this is direct mechano-electrical transduction inside the cell. That's upstream from many different signaling events that, as I said, control cell biochemistry and gene expression. But do cells really use tensegrity? And believe me, I had a lot of critics about this for many years, still do actually. And so the first thing we did, more technique development, I collaborated with Eric Mazur, who had developed femtosecond lasers. Femtosecond laser creates the heat of the sun, but for an incredibly short time, sort of a millionth of a billionth of a second. But we focus it through a microscope objective, so it's focused within a 300 nanometer voxel or cube. So you don't bleach, you vaporize. Some article in the press called it a James Bond laser. And what you can see here is that this is a fluorescent labeling of actin with green fluorescent protein in a living cell. In a living cell, we make a hole punch with that laser, and the circle becomes oblong, meaning like a rubber band, there was pretension, it was under tension, and it stretched. And when you cut the whole one on the right, it's like a muscle being cut from its insertion sites on bone. It pulls back. But nothing else moved. Now, this got critics very excited because they say it shows it's not tensegrity because when I pull on one strut in the model, everything rearranges. But this is on a glass substrate where both ends are anchored to rigid substrate. In our bodies, they're anchored to flexible material. So we put them on a flexible material, and now when we cut one, everything rearranges just like that. Plus, there are little fluorescent beads in the matrix gel, and they retract apart, meaning that the cell and matrix are a pre-stressed tensegrity like a bow and a bowstring. You don't see it, but you cut one, and it splays open. And then finally, people really didn't believe there were compression struts in cells, but microtubules are known to be stiffer, and that if you take them out of the cell, they're straight for millimeters, but in the cell, they have curves, and which I assume was due to buckling. And we could actually confirm that with GFP. This is a heart cell. Every time it beats and contracts, they buckle, and then when it relaxes, they straighten, and that elastic recoil is critical for heart function. If you have too many microtubules, you get heart failure. This was done by Cliff Brangwyn. For those of you who know Cliff, he was at Dresden who won the Breakthrough Award last year for liquid assemblies, being organelles that are actually have no membranes. And he was an undergraduate student when he did this with me. And I work with Dmitry Seminovich, who developed a mathematical model from first principles that predicts many different, both qualitative and quantitative behaviors of many different cell types a priori from basic principles. And this nucleated model, as I said, when I built this, people were absolutely vehement. There is no connection between the cell and nucleus. It's all goopy. But the models predict that this is the case, and in fact, we could show by pulling on integrins, we could make nucleoli become birefringent, which means you're inducing molecular realignment inside the nucleolus, inside the nucleus by pulling on integrins microns away. And we could confirm that there are different filament systems that connect. And recently, this is now an older review, but now this field has exploded. Very definitely, the cytoskeletal filament system connects to structures on the nucleus, and you could even show mechanical force transfer with reporters that read out force. Now, I'm somebody who kind of sees patterns, and I realized that back to my old molecular biophysics class, that molecules are tensegrity. What you see at the left was the most sophisticated tensegrity structure I built when I was 19 years old in my art class. The right is from the cover of Nature, which is a mechanosensitive ion channel. And I think you could see that the alpha helices are stiff regions like a strut, and there are attractive hydrogen bonds that are tensile holding it together. And in fact, we know proteins are pre-stressed because if you clip a protein, it splays open. And this has now been confirmed by many different groups. This is a paper using molecular dynamics simulation of ubiquitin. We measured the network of forces, and we show that it's a tensegrity structure. But in another example of art and science, I have a postdoc who came, Charles Riley, who was trained in molecular biophysics and molecular dynamic simulation, and he worked for Peter Jackson on the Hobbit movie, and he was a professional animator. And so we combined techniques from the entertainment industry that allow you to do multi-scale things. And this is a real depiction, starting from a crystal structure of the dynein motor, put on microtubules in the tail, axoneme tail of a sperm. And it goes down to atomic resolution in the, I'm not sure it's in this video, but it's quite amazing. But we then map out the forces, and you could see tensegrity at the molecular level, the multimolecular, and we could predict how sperm moved, starting from that function of dynein and its architecture in this with tensegrity at every scale. And then it's true at a higher level in our tissues. I did this for Chanel. I was a consultant for their cosmetics group, and they used my tensegrity model in advertisements to look at the resilience of skin. So in skin, you know, most people focus on the chemistry and enzymes that clip it and remodel it, and then the collagens and the proteoglycans that make up, and things you see in skin creams and lotions when you buy them. But what really gives the resilience to skin are the living cells. If you're dead, your skin is rigid. But if you look at what the cells do, they actually interact with these matrix, these are collagen bundles, and they apply tension on their adhesions. This is a focal adhesion modeled computationally. When it pulls in, it rearranges these and pulls them up and stabilizes them. And this is exactly the structure I built in my art class 30 years before. And as I said, artists have described the human body this way for many, many years. And so this was all described in 1998, which is a very long time ago, a cover of Scientific American, where I basically said, and I laid out that starting from the simplest carbon atoms all the way to organisms, this is a fundamental design principle that guides how nature works. And I've also talked about how this could affect the origin of life, if anyone's interested, and wrote on that afterwards. Okay, so that was sort of the basic work that led me in 2009 to form the Wyss Institute for Biologically Inspired Engineering, which is the idea that engineering has transformed medicine and non-medical world by taking engineering principles and trying to solve major problems. We felt that we've uncovered enough about how nature builds, controls, and manufactures that we could flip the paradigm, and we can now leverage biological principles to develop new engineering innovations. This is what I mean by biologically inspired engineering. And as you saw, in my own team, we've uncovered a lot of biological principles. So I should say we kickstarted with the largest single gift in Harvard's history at the time of $125 million, and we were asked specifically to take on high-risk but high-impact challenges, not do incremental work. And the highest risk that I could think of was that the drug development model is broken. It cost over $3 billion, and this is 5 years ago, to take a drug from discovery at the bench to approval in the clinic. There are many reasons, and most of the time the drugs fail when they go into the clinic. There are many reasons for this. You have to do animal studies. It takes years to complete. There are big ethical issues in terms of animal lives, especially non-human primates. The real problem is that 70% of the time, on average, 99% of the time in certain areas of medicine, the results from the animal tests are wrong when you get to human testing. It doesn't make any sense. So there's been a search for, can we develop safer and more effective drugs faster and at lower cost? So when we started the Institute, we had 5 major platform areas. I headed one myself that developed what we called human organs on chips. And the idea here was to engineer microchips containing living human cells that reconstitute organ-level functions, not cellular tissue, but organ-level functions, to accelerate drug development and replace animal testing, and to advance personalized medicine. Now, why on a chip? Well, those techniques that I mentioned for making microchips from George Whiteside's, over the intervening years, we had started to use them to create what are called microfluidic systems. With the same patterning technique, you can make little tiny channels, less than 1 millimeter wide, that you could have little tributaries coming in to make a bigger river. And because they're so small, there's no turbulence. You only get laminar flow. There's no mixing. And so you could have 2 different colored dyes, and they don't mix. And we started to culture cells in these. But what I realized is that this was like an engineered microvascular network, and I worked on blood vessel formation. And so we kind of combined this all together and created what we called organs on chips. And the first breakthrough published in Science in 2010 was a human breathing lung on a chip. Now, we're not trying to build a whole organ or a part of an organ. What I like to you to think of these as our living three-dimensional cross-sections through a major functional unit of an organ. In this case, the air sac, or what's known as the alveolus. And for those of you not familiar, in the lung, the air sac is we have gas exchange, aerosol-based drug delivery, anesthesia, metastasis, viral infections. And if you look at the electron microscopic level, a thin section through, it's pretty simple. There's air, there's one layer of lining cell called an alveolar cell, a porous extracellular matrix, that egg carton that it's attached to, and then the other side of that matrix is a capillary blood vessel cell, and then blood. But what it doesn't show you is that it's an incredibly mechanically active structure, and we know from respiratory physiology that the breathing motions are absolutely critical for lung function and development. So if I were to try to distill down, like, what are the simplest design principles for an organ, and I mean really simple, what I come down to are two or more tissues have to interface and new functions emerge. That's what an organ is. One of those tissues is almost always a blood vessel because you need oxygen and nutrients. And you need dynamic flow because I believe mechanical force is important. And in this case, you need cyclic breathing motions. So the next video shows you how this works. It's the size of a computer memory stick. It's optically clear, made out of that clear silicon rubber. If you cut it in cross-section, it has 3 hollow channels, each less than 1 millimeter wide. The middle channel is cut into top and bottom by another thin, clear, flexible membrane with pores. We coat that with extracellular matrix proteins, those adhesion proteins. If we put human lung lining cells on top, human lung capillary cells on the bottom, we just recreated what's known as the alveolar capillary interface that I showed you at electron microscopic level. The trick is that we have cyclic suction in the side chambers. It's all flexible, so it stretches and relaxes at the same rate and degree as when you and I breathe. You could then flow medium through the bottom, air through the top and have an air-liquid interface like in your lung. You could have medium with or without immune cells, or even whole blood if you have capillary cells on all sides for a short time. Now, if this were to work, it should mimic an organ-level response. So imagine you have a pneumonia, bacterial infection. In your body, there's a tissue-tissue signaling response. The lining cells put out cytokines, which are inflammatory molecules that activate the endothelial cells. Normally, white blood cells just flow by. When they're activated, they stick, roll, migrate across, and engulf. And that's how you fight off disease. So I'm now going to show you imaging in the device. The little white dots are white blood cells. They're fresh, human. I know they're fresh. We took them out of my postdoc. The blood vessel cells are unlabeled. The airway cells are on the other side of the screen. To begin with, the blood vessels, the immune cells just flow by. Now we have infection on the other side. Now they stick. And now you can do any imaging in the device you could do in vitro or in vivo. So I'm going to show you higher magnification in a second. You're going to see one white blood cell there. About here, it's going to find a space between 2 unlabeled capillary cells, goes under them, finds the matrix-filled pentagonal pore, goes out of focus. I'm now going to show you come out the other side by phase contrast imaging. And now I'm going to show you the white blood cells in red because the bacteria are labeled with GFP green and are fluorescent. And you see them being engulfed. So you watch the entire human inflammatory response at high resolution in this little rubber chip. Now, I don't have time to go into all the papers, but this one system has provided proof of principle for human disease modeling, like pulmonary edema, drug toxicity, even when not seen in animals, drug efficacy. We've discovered new targets. There's a drug that moved to phase 2 clinical trials. We've done gene therapy delivery, and it's won numerous awards. I was very proud that it was honored by the World Economic Forum as a top emerging technology. But we won the International Design Award, and we beat out a Frank Gehry building and a Google car that year. But my family's most proud, I'm proud too, that this chip is in the Museum of Modern Art's Permanent Design Collection, which means I'm now a permanent member, which means I get a permanent discount at the cafeteria and the gift shop for life. So that's a few perks you get in science. So we went on and we built many other chips of different functional units. We built a lung airway chip. Now you have airway cells. You have beautiful ciliary motion. If you put fluorescent particles, you can see that they move in a directional way at the same exact rate as mucus moves in our lungs. We made chips. We made a cigarette smoking robot with a cigarette lighter from a car, a Gatling gun with real cigarettes, and it mimics the actual breathing parameters of pulses and pulls and pauses. And we made chips with cells from people that have COPD versus healthy. But this is one data slide where we showed that if you look at a major inflammatory molecule, IL-8, cigarette exposure didn't do much to healthy chips, but if you had chips with cells from patients with chronic obstructive pulmonary disease, COPD, who are ultra-sensitive to cigarette smoke, you could actually see that on the chip. And we could do, nowadays, any sort of sophisticated analysis. This is transcriptomics, but we've done metabolomics, proteomics, glycomics. And in this study, the 3 lanes at the right are 3 different chip responses to cigarette smoke. The 9 at the left are a clinical study of healthy people who smoke cigarettes. We were really excited that the bottom 3/4 versus the top 3/4 look pretty similar. The reviewers turned it down because ours were too similar to each other. And then we realized that we could do this study better than a human study because all these changes are due to cigarette smoke. So they're matched comparative modeling of the same patient before and after stimulus. And these patients have had different work history, different living history, et cetera. So what this made us realize is that organ chips provide a window on molecular scale activities inside living cells within a relevant tissue and organ context. And so what this means is it can enable new mechanistic insights in drug discovery, as well as provide ways to experimentally test predictions immediately in a human relevant model. And so about 5 or 6 years ago, both NIH and DARPA funding agencies in the States asked us, could we take our lung chips and make models of potential pandemic respiratory viruses? But the focus was on influenza at the time. So in this picture, this is GFP influenza virus infecting the cells. But what's nice is you can measure the host response to infection, which you can't do with cells on a dish. I don't know why this is going. This is, these are inflammatory cytokines. This is a cytokine storm you heard about in COVID. Many different cytokines with different viruses, and we match the level of inflammation that is seen in patients on these chips. We even did human-to-human transmission of virus, where we could see evolution of viral variants of concern, if you like. We infected one chip with virus in the presence of drug that was only 90% effective. And then a couple of days later, we take a droplet of fluid through the airspace and cough it, if you like, to another chip with the same drug. And we keep doing this until we get virus that's resistant to drug. With a drug called amantadine, it happened in 8 passages. With Tamiflu, the lead drug, it took 25 passages, but we still got variants of concern. What was more amazing is that when we genome sequenced, we found all of the known mutations that are responsible for resistance seen in human patients, plus ones no one has ever seen before. So you might be able to use this to predict what's coming down the pipe. And then COVID hit. And so I had 2 postdocs from China. They were following this on social media. The day after they published the genome sequence for the the protein sequence for the spike protein that mediates binding, they made pseudotype viruses because we don't have a high security lab, so we could study it in our lab. And they tested 30 drugs in just cells in a dish that they did in their old grad school lab that might have effects against COVID because it worked on prior related viruses. And out of those 30, 8 were active in the cells on the dish, of which 2 were hydroxychloroquine and chloroquine. This is February 2020. But then we tested it in our chips, and hydroxychloroquine and chloroquine had no activity, which has turned out to be true. And we found another related drug that actually showed activity, and that drug moved to clinical trials in 20 sites across Africa, way before we ever got the paper published, I should add. More recently, we've done our lung chip with or without breathing motions, and amazingly, we found that breathing suppresses viral infection significantly. And we mapped out how that works. I'm not going to go into the details, but we found that existing drugs that can suppress the cytokine storm, which is like the major part that causes death in patients. And that drug, the company that makes it has moved that to clinical trials as well. This was all in the last couple of years. We've made chips of every organ. So this is an intestine chip. Intestine, you get intestinal villi in your body. We get those on chip. For the first time, we can put microbiome in direct contact with human cells from multiple days in culture. And interestingly, we found that physiological peristaltic-like motions suppress bacterial overgrowth, which when I went to med school, we learned that when a patient has surgery and has anesthesia, you want to get them eating as soon as possible, because if you don't, they can get what's called ileus, when there's no motion of the intestine and they get bacterial overgrowth. Everyone thought that was because you're not eating, there's no fluids, but we had fluid control, it was the mechanical deformations. And as I said, we can now literally take stool specimens and put them on these chips and study host microbiome interactions. We made a bone marrow chip where we take cells from blood, put them in a gel in one channel. We have blood vessel cells, and we get normal white and red blood cell formation over a month. And a pharma company, AstraZeneca, came to us because they had a drug in clinical trials that had very weird toxicities in that the same dose over 2 hours or 2 days gave totally different toxicities. One gave neutropenia and anemia, which is low white and red blood cells, or, oops, I knew that was going to happen. Don't worry about it. Or neutropenia, which is just white blood cells. And this is something that anybody who does research in a lab usually bathes the cell in drug. Patients get drugs at different dosing, and the drugs go up and down over time, and it's very important that you give the right dosing. We had the measurements in their patients in gray. We could, because we have flow, we could precisely mimic what is known as the pharmacokinetics. 2 hour went up high, came down low, 48 high for a long time, and then dropped. And with that, we could mimic that toxicity precisely. And then with the same chip, we could show that we can carry out personalized medicine and model a rare genetic disorder. We got blood cells from children with a rare disorder known as Shwachman-Diamond syndrome, SDS, who don't have normal blood cell formation. When we made their chips, there was less cellularity just by looking at microscopy, and we could show less white and red blood cells. And we even identified that was a molecular defect no one had identified before, so we got new insight into this disease using this approach. And then just in December, Emulate, a company that I formed that sells these chips and instruments to run them, published a paper where they made 870 human liver chips that had 4 different primary cells from human, the liver hepatocyte, the liver sinusoidal endothelial cells, Kupffer cells, and stellate cells. These are the 4 cells that make up your liver. And they tested 27 drugs where they already knew the results in animals and humans, and most of them had been wrong. And the net sum here is that the chip had 87% sensitivity and 100% specificity in terms of predicting human toxicity of liver, which was 7 to 8 times better than animal models. And this paper had an economic model, talking about other interdisciplinarity, where just this one model should save $2 to $3 billion a year to the pharmaceutical industry by preventing failures later on. And as I said, you know, we've made many different chips. I mean, any organ we've done. But when I first presented this, I had a review article where I said, because we have a blood vessel line channel and a tissue channel, we might be able to make a human body on chips, where you can imagine putting an oral drug through the lumen of the gut chip, watch it be absorbed, see it metabolized by the liver chip, peed out by the kidney chip. Do you have heart toxicity? What does it do to bone? And we actually did this. We don't use tubes. We use a robotic sampler to take 1 drop to go from chip to chip, but 1 drop goes to the chip, 1 drop goes for analysis, like for drug levels. And in this one published study, we had gut, liver, kidney, heart, lung, blood-brain barrier, brain, skin, bone marrow, and pancreas. Kit Parker, also at the Wyss, did the heart and brain chips. And we could keep this alive and functional and see interactions for a month. But the reason we were asked to do this was, could you predict drug pharmacokinetics? Could you predict, before you get to a clinical trial, what dosing regimen you should start with, which could shorten the whole thing, using these chips? And they do it in animals, but it's very different than human. And so here we have a gut, liver, kidney chip, and then we have a little mixing reservoir, so we could take a sample like you're taking a peripheral blood sample. And we added this drug, nicotine, an oral drug like, you know, chewing tobacco or nicotine gum, through the oral part, the lumen of the gut, and we measure its level at every chamber. At the bottom, we do a drug called cisplatin. It's a cancer drug, which is IV, so we put it into this reservoir that mixes to all of them. And again, without going into details, the colored dots are computational predictions, which we did with a company called CFDRC, that mathematically scale this little teeny chip to the mass of each organ and the blood flow rates. And with that, we could quantitatively predict the drug pharmacokinetics of 2 different drugs with the same model. And so to end, you know, what this means is it really can change how we do drug development. Right now, big pharmaceutical companies will spend millions of dollars to do a big clinical trial. They almost always fail. Then they have statisticians do search, is there a genetic subpopulation, a small group that may have responded better? And if they're lucky, They'll do a targeted trial with that group, and if they're lucky, they get approved for a narrow application. With this sort of technology and stem cells, iPS cells, organoids, you can imagine doing, let's say 100 women who have COPD, who are of the same genetic background, find the optimal drug for them, look at their kidney and liver, look to minimize toxicity, choose the most likely candidates to work and do a small trial just with that group and get approved for a narrow application faster, safer, quicker. You could also do things like old versus young, or women versus men, or pregnant women, or things that are rare genetic disorders, or lethal radiation, things you cannot do easily clinically. And I'm very excited that in December of this year, in America, they passed the FDA Modernization Act, which literally says that the FDA can now consider data from human in vitro systems like organs on chips mentioned in it in lieu of animal models, which we really hope will change the way things are done. As a Harvard faculty member, I have to disclose that you can't believe anything I said about organs on chips because I'm a member of the board, chair of the Scientific Advisory Committee and Hold Equity and Emulate, which sells these around the world, and I know there are people at Heidelberg who have them. And again, I could not do this without an incredibly multidisciplinary team that has every sort of expertise that you can imagine. For those of you, I guess there are two messages that I end with here. One is, I think mechanical forces are as important as chemicals and genes. And the other is a quote I love from Buckminster Fuller, which is, nature has no separate departments of biology, physics, chemistry, or art. It is the truth, yet somehow we still teach it that way. And with that, please come to the website if you want to learn more. And thank you so much for this opportunity. I really appreciate it.
Thank you very much, Donald Ingber, for this fascinating lecture in which you gave us an insight into your personal discovery journey, which started with very fundamental insights and ended with applications that help humans to have a better life. Thank you very much. As a sign of our gratitude for your interdisciplinary contribution to Marsilius-Kollegs, we have a little ritual included in this lecture. We have a certificate and a medal, a Marsilius medal for you. And I will first read the certificate to you and then present you with the medal. So the certificate says, the Marsilius Medal is presented to Professor Donald E. Ingber, MD, Ph.D., by the Marsilius-Kollegs of Heidelberg University for his lecture on the topic Bridging Science, Engineering and Art, from Mechanobiology to Human Organs on Chips, and his contribution to the dialogue between scholarly cultures. The date of today and our names. So. And here comes the medal, which is in a case that is hard to open.
I think he wants
so you need many hands for this.
Yes. It's good in the airport security, it'll be interesting.
Thank you very much.